So, what if the very building blocks of life were actually, I don't know, secret agents, doing far more behind the scenes, shaping our existence in ways we rarely even acknowledge?
Yeah, it sounds a bit like sci-fi, doesn't it?
It does. But today we're diving deep into the microscopic world of amino acids.
Mm.
And you're about to discover they are, in fact, biological maestros, really, performing this staggering array of vital functions far beyond just, assembling proteins.
Welcome, everyone, to the deep dive. And today, we're going to truly master the intricate biochemical landscape of amino acid metabolism. Our guide for this exploration, it's primarily Chapter 30 of Harper's Illustrated Biochemistry.
A fantastic resource.
Oh, absolutely. It really delves into the fascinating conversion of amino acids into this diverse range of specialized, absolutely crucial products. And this isn't just about, reading a chapter. It's about internalizing these insights, getting that professional level grasp.
Exactly. And our mission for you, our listener, is to move beyond that foundational, almost simplistic definition of amino acids as just protein components.
Right.
We're going to uncover their hidden vital roles in, everything, from the lightning-fast communication in your nervous system to the intricate dance of energy storage and even the body's sophisticated detoxification pathways.
Yeah.
So by the end of this deep dive, you won't just know what amino acids do. You'll have, hopefully, a professional-level understanding of how critical these molecules are to the body's intricate choreography. It should let you see the biochemical world with, new eyes.
Absolutely. Let's begin this journey, then, to truly understand these cellular masterminds. The underestimated players. Amino acids beyond proteins.
Okay, so when most of us first learn about amino acids, our minds immediately jump to proteins.
Understandably so.
And that's right, isn't it? They are the foundational building blocks, the molecular Lego bricks, if you like, for every protein, from enzymes driving reactions to structural bits holding cells together.
Exactly. That's the classic view.
But to truly grasp their profound biological significance, we need to recognize that their roles extend, dramatically beyond this primary function. They're far more than just construction materials.
You've hit on a crucial point there. The biomedical importance of these lesser-known functions is utterly profound. It's not just about the structures they build, but what they become. And perhaps even more fascinating, how they regulate these vast networks of cellular processes. Think of them as incredibly versatile molecular chameleons.
Okay.
Constantly transforming, adapting their chemical identity to meet the body's diverse, ever-changing needs. From milliseconds to, years, to truly understand biochemistry, you need to appreciate this dynamic adaptability.
And a particularly fascinating aspect of this versatility comes from something called post-translational modifications. Now, this isn't just a simple tweak, is it?
No, not at all.
It's where a protein isn't just made and put to work. It's made, and then certain specific amino acid residues tucked within that protein structure, they get chemically altered.
Right, like a customization.
Yeah, a custom modification, a chemical stamp, which lets them perform very specific, often incredibly intricate functions. It's like a precision upgrade after the initial build. What's a clear example of this in action?
A prime example, and one with massive health implications, is the carboxylation of glutamate.
Okay.
This specific modification forms a new residue called GABA-carboxyglutamate. Now, what's critically important about this seemingly small chemical addition? Acarboxyglutamate is absolutely essential for binding calcium ions, Ca2 plus Asu.
Calcium binding. That sounds important.
It's involved in virtually every major physiological process. We're talking blood clotting cascades, the very ability of your blood to stop bleeding.
Wow.
The intricate mechanics of muscle contraction, rapid nerve impulse transmission, and of course the fundamental process of bone mineralization, giving your skeleton its strength.
So without this little modification,
Without this precise post-translational carboxylation, many of these fundamental processes would simply fail. It leads to severe clinical consequences. It's a perfect illustration. Minute chemical change, monumental biological function.
that's truly staggering for something so chemically specific. And it's not just glutamate that gets these crucial modifications, right? We also see proline being hydroxylated.
Exactly. Proline hydroxylation forms 3 and 4 hydroxyproline residues within proteins. And why is this modification so incredibly vital?
Tell us.
It comes down to structure, specifically the structure of collagen.
Collagen, the abundant one.
The most abundant protein in your entire body, yeah. It's the primary structural component of your connective tissues, skin, tendons, ligaments, cartilage. Its stability is paramount.
Right.
And these hydroxylations, adding the hydroxyl group, provide the necessary cross-linking and rigidity to the collagen fibers. Without them, collagen would be unstable. Your tissues would literally fall apart.
Like rebar in concrete.
Exactly like rebar in concrete. A perfect analogy. And building on that, the hydroxylation of lysine forms 5-hydroxylysine.
Okay.
This 5-hydroxylysine then undergoes further modification and cross-linking, vital for stabilizing those maturing collagen fibers, making them robust, resilient. So these modifications aren't minor tweaks. They are absolutely fundamental to how our tissues maintain strength, integrity, functional elasticity. They ensure our very form holds together.
So we've covered amino acids as building blocks, and then as parts of proteins getting these vital modifications after assembly. But the story gets even bigger, doesn't it? They're also transformed into entirely new molecules, not even proteins.
Indeed. And this broadens their scope dramatically. They are chemically reinvented.
Okay.
Now consider heme, for instance. Heme is this complex ring-shaped molecule. It's primarily synthesized from the amino acid glycine and a related compound, succinyl-CoA.
Right.
Once formed, heme becomes the vital oxygen-binding component of hemoglobin.
The protein in red blood cells.
Exactly. Responsible for carrying oxygen from your lungs to every single cell. It's why your blood is red. Without heme, oxygen transport fails. Life, as we know it, couldn't exist. So a simple amino acid contributes to arguably one of the most critical functions.
And then there are purines and pyrimidines, the core components of DNA and RNA.
The very blueprint of life.
Amino acids are directly involved in making those two.
Directly involved in their synthesis, yes. Plus, think about adenosine triphosphate, ATP.
The energy currency.
It's a purine derivative. ATP powers nearly every cellular process. So amino acids are directly linked to genetics and energy production at the most foundational, inescapable level. It's hard to overstate their importance here.
Absolutely.
Yeah.
And their influence doesn't stop there. Beyond these, amino acids also serve as direct precursors for various hormones.
Hormones, right. Regulating metabolism, growth.
Countless functions. But where it gets really compelling for many people, connecting directly to our everyday experiences, is their pivotal role in producing neurotransmitters. Yes, neurotransmitters, the chemical messengers of the brain and nervous system, dictating mood, movement, thoughts, perception, sleep cycles. Everything.
One familiar one is histamine, usually associated with allergies, right? Itchy eyes, runny nose.
That's the common association, yeah. But histamine also plays a central role in gastric secretion, helps with digestion. And it's present in virtually all tissues, showing its widespread biological significance.
Interesting.
And get this. Its concentration in the brain's hypothalamus even shows circadian rhythm-dependent variations. Hints at involvement in our internal clock and sleep-wake cycles.
Wow. And then there are others derived directly from amino acids.
Oh, yes, absolutely critical ones. Think about aminobutyrate, or GABA. That's primarily an inhibitory neurotransmitter, the brain's internal brake.
The brake, okay.
Then there's 5-hydroxytryptamine, much better known as serotonin, influences mood, sleep, appetite.
Serotonin, yeah. And finally, the powerful catecholamines. Dopamine, norepinephrine, and epinephrine. All of these vital chemical messengers, essential for virtually every aspect of brain function, are directly synthesized through specific transformations of amino acids. It's a direct link between, well, what you eat and how your brain functions.
What's truly fascinating here, and clinically relevant, is the immense medical impact. So many drugs for neurologic and psychiatric conditions, depression, anxiety, Parkinson's.
A huge number.
They work by directly altering the metabolism of these very neurotransmitters. It really makes you wonder, how deeply do these drugs interact with the underlying biochemistry? It's not just a simple on-off switch, is it?
Not at all. It's a remarkably complex interaction. Understanding it is paramount for drug development. These drugs often target specific enzymes, the ones involved in making the neurotransmitter, or breaking it down, or taking it back up into the neuron.
So fiddling with the levels.
Exactly. The goal is usually to increase or decrease the amount of a signaling molecule in the synaptic cleft, where neurons talk to each other. To design truly effective, targeted therapies and minimize side effects, you absolutely must have a professional-level understanding of the specific amino acid precursor and the entire intricate metabolic pathway it undergoes. It's a delicate balance. Small alterations can have profound systemic effects. 2. The Alpha-Amino Acid Hall of Fame. Specialized roles. Detail.
All right, let's get specific then. We're moving into what we're calling the Alpha-Amino Acid Hall of Fame. We'll detail these specialized vital roles of some key players. First up, Alanine. Often seen as basic, but it's really a metabolic carrier.
Alanine plays an essential role, yes. It carries ammonia and the carbon skeletons derived from pyruvate. Primarily, it transports these from skeletal muscle, especially during intense exercise, to the liver.
Part of that Cori cycle, or glucose-alanine cycle.
Exactly. It's critical for maintaining glucose homeostasis. In simple terms, muscle breaks down protein or goes anaerobic, needs to get rid of waste nitrogen and send carbon fuel back to the liver. The liver detoxifies the nitrogen, makes new glucose. Alanine is the shuttle.
Keeps blood sugar stable, gets rid of toxic ammonia.
Precisely. And alanine, along with glycine, makes up a major fraction of the free amino acids circulating in your plasma, highlights its abundance and constant movement as this metabolic shuttle.
Next, arginine. Truly the versatile contributor. This one seems involved in so many different crucial pathways.
Arginine is incredibly versatile, almost a biochemical multi-tool. Besides being in proteins, it's a vital carrier of nitrogen atoms in urea biosynthesis, how we safely ditch excess toxic nitrogen from protein breakdown.
The urea cycle.
Right. Its unique guanidino group, that nitrogen-rich structure, gets directly incorporated into creatine.
The muscle energy buffer.
The key energy buffer, providing rapid bursts of ATP. And after it's converted to ornithine in the urea cycle, arginine's carbon skeleton serves as a precursor for polyamines like putrescine and spermine,
which are for cell growth.
Crucial for cell growth and proliferation. We'll touch on those again with methionine.
And then there's nitric oxide production. Now we hear a lot about that. Cardiovascular health, brain function. How does arginine fit in?
Precisely. The enzyme nitric oxide synthase, its complex, needs multiple cofactors, catalyzes this remarkable reaction, converts one nitrogen from arginine's guanidine group directly into nitric oxide.
Just like that.
And what's absolutely fascinating here is NO's incredibly diverse impact. It's not just a gas, it's a powerful intracellular signaling molecule. It acts as a neurotransmitter in the brain.
Okay.
A potent, smooth muscle relaxant which explains roles in erectile function, gut motility, and a powerful vasodilator. It widens blood vessels.
Critical for blood pressure and flow.
Absolutely. Regulating blood pressure, blood flow to the heart, brain, everywhere. Its widespread influence underscores its critical role in vascular health, nervous system function. This single molecule from arginine orchestrates a vast array of physiological responses.
Quite a resume for a tiny gas molecule.
Yeah.
And speaking of energy, you mentioned creatine phosphate. Arginine plays a similar energy storage role elsewhere too.
It does. Arginine phosphate, found in invertebrate muscle, functions as a phosphagen, an energy storage compound analogous to creatine phosphate in mammals.
Same strategy, different molecule.
Highlights a conserved evolutionary strategy for rapid energy buffering. Quick bursts of activity, whether it's a sprinting or a crab escaping, it really links arginine to fundamental energy homeostasis across species, shows its ancient vital role.
Truly global impact.
Right.
Okay, moving on to cysteine, our sulfur specialist. What makes it so special?
Cysteine is unique because it contains that sulfur atom, makes it a key player in many sulfur-containing compounds. Notably, it participates in coenzyme A biosynthesis, reacts with pantophenate, a B vitamin, to form 4-phosphopanthenol cysteine.
And coenzyme A is central.
Absolutely central to metabolism, involved in countless reactions. Fatty acid synthesis, fatty acid oxidation building and breaking down fats, the citric acid cycle, the hub of energy production. It's a metabolic linchpin, a universal adapter for carrying acyl groups.
Everywhere in metabolism.
Sure.
And cysteine also forms taurine, which helps make bile acids.
Yes, the conversion of cysteine to taurine is a multi-step enzymatic process. Once formed, taurine has a critical role in the liver. It displaces the coenzyme A from choline-CoA to form taurocolic acid.
A key bile acid.
A key bile acid. And bile acids are essential for digesting and absorbing dietary fats and fat-soluble vitamins, A, D, E, K, in your intestines. Without proper bile acid synthesis, you can't effectively break down and absorb these crucial nutrients, leads to deficiencies. So cysteine sulfur is ultimately vital for digestive health.
Okay, let's turn to glycine. Often seen as simple, but it's a powerful detoxifier and a fundamental builder.
Glycine is indeed instrumental in the body's detox systems. Many relatively apolar, fat-soluble metabolites, things the body needs to get rid of, like benzoate from food or numerous drugs and their breakdown products, are converted into water-soluble glycine conjugates, like hippuric acid. This conjugation process significantly increases water solubility, making them much easier to excrete in urine.
Makes them easier to flush out.
Exactly. It's a crucial pathway for clearing xenobiotics, foreign chemicals, and various other compounds with carboxyl groups. prevents accumulation and potential toxicity. Think of it as a chemical expel me flag.
The body's internal water-soluble flag, but it's also a fundamental builder, right? Beyond just proteins.
Absolutely. Glycine is a component of creatine, that essential energy reserve.
We keep coming back to creatine.
It's important. More profoundly, glycine's nitrogen and its alpha carbon are directly incorporated into the pyrrole rings and methylene bridge carbons of heme.
The oxygen carrier in hemoglobin.
Right. And going even deeper, to the core of life, the entire glycine molecule supplies atoms 4, 5, and 7 of the purine bases, the A and G, in your DNA and RNA.
Wow.
Emphasizes its foundational role in constructing genetic material and the universal energy currency, ATP glycine. A silent partner in your genetic identity and energy supply.
Moving on to histidine, the allergic responder, key in gastric secretion and a tissue buffer. Surprising breadth of roles here.
Histidine's most widely known non-protein fate is its decarboxylation to histamine via histidine decarboxylase.
Histamine, right.
As we mentioned, it's a powerful biogenic amine, prominent in allergic reactions, redness, swelling, itching due to local immune responses, but also critical roles in gastric secretion, stimulating stomach acid release. Found in virtually all tissues, widespread influence.
And the circadian rhythm connection in the brain.
Yeah, its concentration in the hypothalamus varies with the circadian rhythm, suggesting roles in sleep-wake cycles, maybe appetite regulation too.
And there are other histidine-containing compounds too, not just free histamine. What do they do?
Yes, very important ones. Compounds like carnosine, that's bolanyl histidine, and homocarnosine, aminobutyryl histidine. Major constituents, abundant in excitable tissues like brain and skeletal muscle.
What they thought to do there.
Roles in regulating calcium, acting as powerful antioxidants. We also get dietarily derived ones like ergothioneine and anserine, shows the diverse metabolic fates. Clinically, low urinary levels of 3-methylhistidine, a breakdown product, are seen in Wilson disease.
The copper disorder.
Right. A subtle metabolic marker offering a glimpse into the body's copper handling.
Fascinating connections. Now, methionine, the universal methyl donor and polyamine source. Like cellular superglue, adding methyl groups everywhere.
That's a great analogy. Methioninine's major non-protein fate is conversion to S-adenosylmethionine, or SAM. SAM is truly the principal source of methyl groups in the body, essential for countless methylation reactions.
Methylation, like adding a chemical tag.
Yeah, exactly. Adding a small tag, methyl group, to molecules, which can dramatically change their function. Critical for DNA replication and repair, gene expression, turning genes on and off, neurotransmitter synthesis, lipid metabolism, detoxification, the list goes on.
How is it made?
Biosynthesis is from methionine and ATP, catalyzed by methionine adenosyltransferase, or MAT.
And there are different forms of this MAT enzyme. Does that give the body some flexibility?
Yes, 3 human MAT isozymes. MET1 and AT3 are mainly in the liver, MAT2 in other tissues. This distribution is key. Interestingly, even if hepatic MT13 activity is severely decreased, leading to hypermethionemia, high methionine in the blood, if there's some residual activity plus normal AT2 activity, a high tissue concentration of methionine can actually ensure adequate SAM is still synthesized.
So backup systems.
Indicates metabolic flexibility or redundancy. The body has ways to ensure production of this absolutely crucial molecule. Testament to resilience.
So the body can adapt to keep making this crucial methyl glue. And SAM also contributes to polyamine biosynthesis. You mentioned those earlier. What are polyamines again, and why are they important?
They do. Critical pathway. After SAM is decarboxylated by methionine decarboxylase, 3 carbons and the alpha-amino group of methionine are used to make polyamines like spermine and spermidine.
And their role?
Absolutely crucial in cell proliferation and growth. They act as growth factors for cultured mammalian cells, essential for tissue development, repair. They also stabilize intact cells, organelles like mitochondria, membranes. Structurally, because they have multiple positive charges, they readily associate with and stabilize negatively charged molecules like DNA and RNA.
Helps hold our genetic code together.
Essential for maintaining the integrity and function of our genetic material. Pharmacologically, high doses can be hypothermic, lower temperature, and hypotensive, lower blood pressure, highlighting their potent biological activity.
Huge impact for these molecules. Next, serine, a structural and signaling scaffold.
Serine is remarkably versatile. Involved in sphingosine biosynthesis, a critical component of many lipids, especially in nerve cell membranes, the myelin sheath.
Insulating nerves.
Right. It's also a crucial nucleotide precursor, provides carbons 2 and 8 of purines and the methyl group of thymine, a pyrimidine base in DNA.
So directly contributes to building blocks of DNRNI.
Directly. And a significant clinical connection. Serine condenses with homocysteine to form cysteine via cystathionine bisynthes. Genetic defects in this enzyme cause homocystinuria.
A serious metabolic disorder.
Very serious. Can cause intellectual disability, skeletal problems, increased risk of blood clots. And interestingly, serine, not cysteine, is the precursor for peptidyl selenocysteine.
A unique amino acid.
Found in selenoenzymes, vital for antioxidant defense, thyroid hormone metabolism. Really highlights how one amino acid can have such a wide ripple effect.
All right, let's move to tryptophan, famously linked to neurotransmitters and sleep regulation.
Yeah.
The whole Thanksgiving turkey sleepiness thing. Mostly a myth, right? But how does its biochemistry actually work?
Good point about the myth. Tryptophan's actual pathway is fascinating. It gets hydroxylated by tryptophan hydroxylase, mainly in the liver, forming 5-hydroxytryptophan.
Okay.
This intermediate then gets decarboxylated to form serotonin, 5-hydroxytryptamine. Serotonin itself is a potent vasoconstrictor, stimulates smooth muscle contraction, plays widespread roles not just in the brain, but significantly in the gut.
Right. Most serotonin is in the gut, influencing digestion.
Exactly. And how is it broken down? Catabolism.
Yeah, and the clinical implications of messing with that.
Serotonin catabolism starts with deamination to 5-hydroxyanyl-3-acetate, catalyzed by monoamine oxidase, MAO. MAO acts as a critical brake on serotonin's activity. This is clinically profound. Drugs like aproniazid, an antidepressant, work by inhibiting MAO.
So that prolongs serotonin's action.
Exactly, leading to psychic stimulation. This directly answers your earlier question. Inhibiting a catabolic enzyme increases and prolongs the presence of the signaling molecule in the synapse, enhancing its effect.
Blocking the breakdown boosts the signal. Fundamental for many psychiatric meds.
Precisely. In conditions like carcinoid, a type of tumor where cells overproduce serotonin, the excess serotonin gets metabolized. Urinary levels of its breakdown products, like N-acetylserotonin glucuronide and the glycine conjugate of 5-hydroxyindoleacetate, become important diagnostic markers. Helps clinicians monitor the disease.
I see.
MAO also metabolizes other compounds, like 5-methoxytryptamine, to their acids. Shows its broad role in detoxifying biogenic amines.
And then there's melatonin, the sleep hormone. How does tryptophan lead to that?
Melatonin is formed directly from serotonin. N-acetylation of serotonin, then O-methylation in the pineal body, that small gland in the brain.
Pineal gland, right.
Once circulating, melatonin is taken up by all tissues, including the brain, but its rapidly metabolized hydroxylation, then conjugation with sulfate or glucuronic acid.
So it doesn't hang around too long.
Helps in efficient excretion, ensures its effects are transient, properly regulated for our sleep-wake cycle. Tryptophan also yields other metabolites. Kidney, liver, even gut bacteria can convert it to tryptamine, then indole-3-acetate. Principal normal urinary catabolites are 5-hydroxyindoleacetate and indole-3-acetate. So a complex and far-reaching journey for tryptophan.
Next up, tyrosine. Precursor for catecholamines, fight or flight, and also thyroid hormones, directly linked to stress response and metabolism.
Indeed. Neuronal cells, especially in the brain and adrenal glands, convert tyrosine into the powerful catecholamines, epinephrine, adrenaline, and noradrenaline.
How does that pathway work?
Starts with tyrosine hydroxylation forming DOPA. Now, DOPA is also an intermediate in melanin formation skin pigment, but different enzymes handle the hydroxylation in melanocytes versus neurons.
Context matters.
Very much so. From DOPA decarboxylase needs vitamin B6, forms dopamine, critical for reward, motivation, motor control.
Dopamine, okay.
Then, subsequent hydroxylation by dopamine oxidase yields norepinephrine, vital for alertness, stress response. Finally, in the adrenal medulla gland on the kidneys,
Right.
an enzyme called phenylethanolamine N-methyltransferase uses SAM, our methyl donor, from methionine to methylate norepinephrine, making epinephrine, the quintessential fight-or-flight hormone.
A whole complex chain reaction from one amino acid dictating our primal responses. And it's also a precursor for thyroid hormones, regulating our entire metabolism.
Yes, tyrosine is also the direct precursor for triiodothyronine, T3, and thyroxine, T4. These hormones are absolutely crucial for regulating metabolism, growth, development throughout the body.
Everywhere.
They influence virtually every cell and organ, heart rate, body temperature, brain function, energy levels. So one amino acid, tyrosine, stands at the nexus of rapid neural signaling and long-term metabolic regulation. Underscores how tightly integrated these pathways are.
Now let's talk about something that acts like a master on-off switch.
Yeah.
Phosphorylated amino acids. Adding phosphate to serine, 309, or tyrosine within protein.
Here's where it gets really interesting. Moves beyond synthesis into sophisticated cellular control. Phosphorylation and dephosphorylation, adding and removing phosphate on specific cyral, thrynyl, or tyrosyl residues, play key, dynamic roles in regulating the activity of countless enzymes, especially those in lipid and carbohydrate metabolism. Beyond that, they are central to proteins in complex signal transduction cascades.
The cell's communication networks.
Exactly. Allowing cells to respond to external signals like hormones, growth factors. These seemingly small modifications, adding or removing phosphate, act like molecular on-off switches or even dimmer switches.
Controlling crucial processes.
Fundamentally altering a protein's function, its interactions, its location, thereby controlling vast networks of biochemical reactions. It's how cells receive messages, respond to stimuli, grow, divide, even know when to die. It's the very language of cellular regulation.
The master control panel for the cell. Okay, next, sarcosine, or N-methylglycine, a one-carbon unit source. What's its role? Seems less famous.
Its biosynthesis and catabolism happen mainly in the mitochondria, the cell's powerhouses. Sarcosine can be formed from dimethylglycine via dimethylglycine dehydrogenase. This needs reduced tyrosine pentaglutamate, a B vitamin cofactor for moving single carbon units.
Right.
Traces can also arise from direct methylation of glycine using SAM, catalyzed by glycine and methyltransferase.
So formed and broken down in the mitochondria. What about its breakdown, and why are these one-carbon units so important again?
Catabolism back to glycine is by sarcosine dehydrogenase, also needing that reduced tyrosine pentaglutamate cofactor. These demethylation reactions removing methyl groups that form and degrade sarcosine are important sources of those one-carbon units.
And they're vital because?
Crucial for various metabolic processes. Synthesis of nucleic acids, drRNA, other amino acids like methionine. They're building blocks for methylation, which, as we saw with SAM, is essential for gene regulation, detoxification, and the FADH2 generated in these reactions. Reoxidized via the electron transport chain. Directly links the seemingly niche metabolism to the grand process of energy production in the mitochondria.
Finally, in this alpha amino acid section, creatine and creatinine. Energy buffers, but also useful clinical markers.
Creatine phosphate, major energy reserve, abundant in muscle and brain, provides quick, available ATP for immediate energy needs. Sudden, intense activity, like a rapidly rechargeable battery.
Got it. And creatinine.
Formed in muscle from creatine phosphate. Irreversible, non-enzymatic dehydration, loss of phosphate. It's a waste product, but a very useful one.
Useful how? What does it mean for us practically, say, in a hospital?
It's a simple, non-invasive way to check muscle mass and, crucially, assess the completeness of urine samples. The 24-hour urinary excretion of creatinine is remarkably proportionate to an individual's total muscle mass.
So it tells you if you got the whole 24-hour sample.
Exactly. Provides a reliable measure of whether a complete specimen was collected. Helps ensure accuracy of other tests relying on total urine volume. A vital quality control indicator. Creatine biosynthesis itself involves glycine, arginine, and methionine. Completed by methylation of granodidoacetate by SAM, highlights the beautiful interconnectedness. 3. The non-alpha-amino acid ensemble. Unique structures, unique functions.
That was a truly comprehensive look at the alpha amino acids. But wait, there's this whole other cast. The non-alpha amino acids. Unique structures, unique functions, even though they aren't directly built into proteins.
That's absolutely right. These aren't typical protein building blocks, but they're present free, or in small peptides, in tissues, performing specialized roles. Examples, beta-alanine, isobutyrate, and beta-aminobutyrate GABA.
Right, GABA.
And beta-alanine is also found combined in coenzyme A and beta-alanyl dipeptides. Not always free-floating.
Let's start with beta-alanine and beta-aminobutyrate. Intriguing because they're products of pyrimidine catabolism, from breaking down bits of our genetic code.
Precisely. Formed during the breakdown of pyrimidines. Alanine from uracil catabolism, aminoisobutyrate from thymine catabolism. When your body recycles old RNA or DNA, these are byproducts.
Okay.
We also find traces of alanine from hydrolysis of doralanyl dipeptides by carnosinase. And boraminoisobutyrate can also come from transamination of methylmalonate semialdehyde. a breakdown product of the amino acid alvaline, shows that intricate web of metabolic recycling.
So not just from genetic waste, also from other amino acid paths. How are they broken down? What's special about their final products?
For valine, initial transamination to malonate semialdehyde. Then CoA transfer from sequential-CoA forms malonyl-CoA semialdehyde, oxidized to malonyl-CoA, finally decarboxylated to the amphibiolic intermediate acetyl-CoA.
Amphibiolic, meaning it can go both ways, building or burning.
Exactly. Acetyl-CoA can be used for building things like fatty acids, anabolic, or broken down in the citric acid cycle for energy, catabolic. Aminoabutyrate follows analogous reactions. Transamination forms methylmalonate semialdehyde, converted to the amphibiolic intermediate succinyl-CoA. Also feeds directly into central energy production. So these pyrimidine breakdown products ultimately contribute to the body's energy currency.
And what if these breakdown pathways go wrong? Problems breaking down the breakdown products.
Disorders of alanine and mannose-butyrate metabolism usually arise from defects in pyrimidine catabolic pathway enzymes, primarily conditions from deficiency of dihydropyrimidine dehydrogenase, DPD.
DPD.
Crucial for the first step in pyrimidine breakdown. Deficiency leads to buildup of precursor pyrimidines. Also highly clinically relevant because DPD metabolizes certain chemotherapy drugs like 5-fluorouracil.
Oh, wow.
Patients with DPD deficiency can have severe, even life-threatening toxic reactions to these drugs. Highlights the link between our own metabolism and drug metabolism.
Now, alanine peptides, the muscle protectors. What are they and why are they so important for physical performance?
Indeed, prominent examples: carnosine and anserine, and methylcarnosine. Several fascinating functions. Activate myosin ATPase, critical for muscle contraction energy use. Roles in chelating copper, enhancing copper uptake, suggest roles in trace elements, antioxidant defense.
But their most famous role?
The alanyl imidazole portion effectively buffers the pH of anaerobically contracting skeletal muscle.
Buffers the acid buildup during intense exercise.
Exactly. Crucial for maintaining performance during intense short bursts when lactic acid builds up, lowering pH. Carnosine buffers this acidity, helps delay muscle fatigue, maintain power output, a direct biochemical link to athletic performance, endurance.
What an athlete feels during a sprint. How are these buffers made and broken down?
Carnosine biosynthesis is by carnosine synthetase, a two-stage reaction. First, enzyme-bound acyladenylate of balanine, then transfer of balanil to L-histidine. Breakdown, hydrolysis back to balanine and L-histidine is by carnosinase.
And there's a disorder.
A rare, heritable disorder, carnosinase deficiency, characterized by carnosinuria carnosine excreted in urine due to the enzyme defect, shows even minor components have significant consequences if metabolism is disrupted.
And homocarnosine, you mentioned it earlier, similar roles.
Present in human brain at higher levels than carnosine. Also synthesized in brain by carnosine synthetase. Interestingly, serum carnosinase, the one in blood, does not hydrolyze homocarnosine. Suggests specialized roles, regulation.
And a disorder there too.
A rare genetic disorder, homocarnosinosis, associated with progressive spastic paraplegia, muscle stiffness, weakness, and mental retardation. Again, severe consequences when specific metabolic pathways are disrupted, especially in the nervous system.
Lastly, in our non-alpha ensemble, aminobutyrate, GABA. The brain's inhibitory brake. Okay, unpack this. Why is an inhibitory neurotransmitter so fundamental? Seems counterintuitive that slowing things down is vital.
That's a very insightful question. GABA functions in brain tissue as an inhibitory neurotransmitter by altering transmembrane potential differences. It makes nerve cells less likely to fire. It slows down or stops nerve impulses.
So it prevents overexcitation?
Exactly. This braking action is critical for maintaining neural balance. Without sufficient inhibition, the brain would be prone to overwhelming electrical activity seizures, severe anxiety, uncontrollable tremors. GABA allows for precise control, fine-tuning of neural circuits, ensures signals are transmitted clearly, efficiently, without chaotic interference, prevents an electrical storm, allows organized thought, controlled movement.
The traffic cop for nerve impulses, keeping things calm and controlled. How is it formed and broken down? Clinical implications.
Formed by simple, crucial decarboxylation of glutamate via L-glutamate decarboxylase. Glutamate itself is a major excitatory neurotransmitter, so this conversion is key, turning an accelerator into a brake.
Accelerator to brake, got it. And breakdown.
Catabolism starts with transamination of GABA, forming succinate semialdehyde. This semialdehyde can then take two paths. reduced to hydroxybutyrate, biolactate dehydrogenase, or more commonly, oxidized to succinate.
Which feeds into the energy cycle.
Right, enters the citric acid cycle, fully metabolized to CO2 and H2O, effectively recycled back into energy production.
And disorders linked to problems with GABA metabolism, given its critical role.
Yes, unfortunately, rare genetic disorders highlight its importance. One involves defective GABA aminotransferase involved in GABA catabolism after release. If it doesn't work, GABA persists too long.
Okay.
Another, more discussed, involves defects in succinic semialdehyde dehydrogenase, responsible for 4-hydroxybutyric aciduria,
What's that?
a rare metabolic disorder characterized by significant amounts of 4-hydroxybutyrate in urine, plasma, cerebrospinal fluid, accumulation of a toxic byproduct because the pathway is blocked.
And the symptoms, treatment challenges.
Patients typically present with mild to severe neurologic symptoms, developmental delay, intellectual disabilities, seizures, movement disorders. Sadly, there's currently no specific, highly effective treatment beyond supportive care.
Wow.
Highlights the huge challenges in managing complex metabolic disorders where a single enzyme defect in a fundamental pathway has such widespread, debilitating, and currently intractable neurological consequences. A stark reminder of the intricate balance needed for healthy brain function. Outro.
Wow. We've truly gone from seeing amino acids as just simple building blocks to, well, orchestrators of incredibly complex biological processes,
Right.
each with multiple, often hidden roles, like a single musical note being a melody, rhythm, or counterpoint, depending on context.
Indeed. To recap quickly, beyond proteins, alpha amino acids do so much. Arginine gives us creatine for energy, nitric oxide for signaling, vasodilation, and contributes to polyamines stabilizing DNA. Cysteine sulfur is vital for coenzyme A, that central metabolic adapter, and for bile acids and digestion.
And glycine is fundamental to heme for oxygen transport, purines for our genetic code, creatine for muscle energy, and a silent hero in drug detox.
Histidine produces histamine allergies, yes, but also gastric secretion, sleep rhythms. Methionine is the universal methyl donor, indispensable for polyamines, genetic regulation, countless other reactions. Serine is vital for lipids, nucleotides, linked to disorders like homocystinuria. Tryptophan gives us serotonin for mood and gut, melatonin for sleep.
Tyrosine, the precursor for critical catecholamines like epinephrine, norepinephrine, stress response, and vital thyroid hormones regulating metabolism, plus phosphorylated amino acids acting as cellular on-off switches, and glycine, arginine, SAM collaborating for creatine.
Then we explored the non-alpha-amino acids, beta-alanine and bepaminosorbulate from pyrimidine catabolism, highlighting how defects cause disorders, valinol dipeptides like carnosine protecting muscles, buffering pH.
And finally, GABA, the brain's essential inhibitory brake. Severe neurological issues arise from defects in its catabolism, like 4-hydroxybutyric aciduria. Truly mastering this gives that professional-level understanding a deeper appreciation for the intricate choreography of biochemistry. Shows how seemingly small molecules, these biological secret agents, have enormous impacts on health, disease, life itself.
It's an incredible interconnected web, isn't it? Every molecule, every pathway, profoundly connected, each playing its vital, multifaceted role.
Absolutely. And this level of detailed understanding, it really opens up profound, exciting questions, doesn't it?
It does, considering how deeply these pathways are intertwined, how one amino acid's fate cascades if a single enzyme is defective. What new avenues might this understanding open for future therapies, especially for currently untreatable neurological disorders where a subtle biochemical imbalance might just hold the key? The more we truly understand these secret lives of amino acids, the closer we get to unlocking new solutions and deeper insights into human health.
A powerful and thought-provoking question to leave you with. Thank you for joining us on this deep dive into the unseen, vital lives of amino acids. We hope you not only feel more well-informed, but are truly amazed by the incredible, intelligent biochemistry at play within you, well, every single second.